Chapter 3 Microbial flora of milk and milk product. Microbial flora of milk and milk product, yeast, mold, and virus. Learning outcomes. Incomplete of this chapter, the learner will have a knowledge of the morphology and type of yeast and mold associated with the milk and milk product, partitioning to spoilage and consumer health. Have a critical knowledge about the role of bacteriophage in dairy industry with special emphasis on this multiplication and prevention of spoilage, especially in fermented milk industry. Yeast. Yeast. Yeast are unicellular, ovoid or elliptical cell, gram-positive, non-motile, and large in size when compared to bacteria. Reproduction is by budding, growth well at 25 to 40 degrees Celsius and can tolerate pH 3.5 and are strictly aerobic. Saccharomyces cerevisiae, used in the brewing and baking industry and in kefir production. Cells are cylindrical to ellipsoidal, occurring singly or in pair, short chain or cluster. Lactose is not fermented. Candida kefir. Candida kefir. Candida kefir. Associated with kefir, buttermilk, and cheese. Produce pseudomycelium, some strain fermented lactose. Candida lactinodense. Turulopsis lacti. Isolated from condensed milk. Lactose non-fermented. Candida pseudotropicalis, referred to as false yeast, ferments lactose with the production of alcohol and carbon dioxide. Associated with yeast or greasy cream and kefir gan. Debromyces hansenii has been isolated from cheese, occurring in short chain and propagation by multipolar budding does not ferment lactose. Chloromyces Vermaxianus, previously known as Chloromyces fragilis, associated with manufacture of kefir and kumis. Cells occurs as subglobose and signally or in pair. Previously known as Chloromyces lactis, associated with yogurt and has been isolated from milk, greasy cheese, buttermilk, and creamy. Cell are amoeboid and will fermented lactose. Molds. Mold. Mold. Molds are complex multicellular organisms, aerobic growing over a wide range of pH. The colony appear cottony or woolly and are generally white, creamy, green, or black. The individual mold is composed of an aggregate of branching protoplasmic thread known as mycelium. Individual threads of the mycelium are termed as hyphae. They are vegetative, concerned with nourishment, or fertile, concerned with asexual or sexual reproduction. Some produce sporangium, which in turn produce sporangiospore, and some candida, which produce candidiospore. Penicillium species. Penicillium species. The Penicillium species usually from blue-green spreading colony. Example: Penicillium roqueforti, which is used in the manufacture of roquefort cheese and blue vended cheese. It is mainly responsible for characteristic flavor and appearance of this cheese. Another species, namely Penicillium combatii, is used in the manufacture of Camembert cheese and Brie cheese. Its colony are pale green in color and have the appearance of typical paintbrush. Spores are produced on specialized structure called conidiophores, and the spores are known as conidiospore. Rhizopus species. Rhizopus species. Rhizopus species are present in foodstuff like bread and dairy products. The mycelium produce stolones, which in turn give rise to rhizoids, root-like and sporangiosperm producing structure. Rhizopus stolonifer is involved in replenishing of gamulous variety of cheese, reproduced by conidiospore, which are borne on stigmata. Aspergillus species. These spoil sweetened condensed milk often, and the mold mycelia and the coagulated casein from characterize buttons. Some species, Aspergillus flavus, Aspergillus parasiticus, are capable of producing aflatoxin G1, G2, B1, and B2 in dairy products. The aflatoxin are elaborated in animal feed as well, which consequently get secreted as M1 and M2 in milk. Aspergillus niger produce black-colored colony. Geotrichum candidum. Geotrichum candidum. This is responsible for yeast flavor in dairy products. It is lipolytic in nature and colony appear yeast-like. They grow on the surface of the sour cream and hydrolyze it. Reproduced by arthrospore, which are cylindrical with rounded cell, and Cladosporium species. Cladosporium species. Cladosporium is deep olive green to black color, often associated with surface discoloration of butter. Contamination of these in the mold dependent cheese varieties give rise to ammoniacal odor. The conidiospore of this species are characteristic. They are truncated sphere with a thickened basal ring around the truncation. Verrucase. A verrucase particle or virion consists of nucleic acid, DNA or RNA that is covered by a protein coat called capsid are symmetrically constructed. Two kinds of symmetric have been recognized: helical cuboidal. Hepatitis virus. Hepatitis virus causes hepatitis, which is generally referred to as jaundice. It is due to the consumption of milk or food contaminated with feces or carriage of this disease. Cowpox virus produce pustules and vesicles on the teeth of cow udder. Poliomyelitis virus belong to the group of enterovirus. It is readily killed by pasteurization. It has tendency to get localized in the CNS, leading to paralysis. Tick-borne encephalitis. Tick-borne encephalitis belong to the genus Palovirus and is responsible for meninge or meningoencephalitis. Its vectors are the tick Aedes racinus and I. persulatus. The disease may be caused due to consumption of unboiled goat milk. Bacteriophage of lactic acid bacteria. Bacteriophage of lactic acid bacteria. Bacteriophage are the viruses that infect bacteria. Many times in cheese industry, starter culture fail, which is due to bacteriophage. Bacteriophage in milk. Bacteriophages in milk. Bacteriophage may be regarded as viruses or ultramicroscopic organisms, especially parasitic on bacteria. The phages are highly specific for bacterial strain. A phage has been numerically the most important cause of slow starter action, and there may be complete cessation of starter growth or failure. A successful phage attack on a starter culture may bring the acid development to a complete halt, and acidity may not develop again until either a fresh strain of lactic streptococci succeed in establishing itself, or possibly a few phage resistant cells from the original strain ultimately resume growth. The phages are highly specific for bacterial strain. They are widely distributed in nature and most evident in the intestinal content of animals. They are too small to be seen under ordinary microscope, which can only be seen in the electron microscope after the application of suitable staining technique, and cannot be removed from a solution by the filter of the type which remove bacteria since their size is significantly less than that of a bacterium. Phage literally mean developing, devouring, just like phagocyte, to swell or eat up gradually. They are widely distributed in nature and most evident in intestinal content of animal, and first is invented by Toward in 1950. Like microorganisms and enzymes, they are destroyed by heating for 30 to 60 minutes at a temperature of 70 to 75 degrees Celsius. However, they are very resistant under favorable conditions and may remain variable after dying. They are destroyed by ultraviolet light but resist dilute HCl2 solution. Certain dyes such as methylene blue may also inactivate phages, particularly when photosensitization. The phage have a head and must have a tail. The head may be isometric or with a hexagonal outline. There are two types of phage, virulent and temperate, which enter a different relationship with the host, the lytic and lysogenic cycle respectively. Multiplication of cycle of bacteriophages. Multiplication cycle of bacteriophages. Lytic cycle. In the lytic cycle, phage multiplication occurs in four main sequential steps: phage absorption, DNA injection, phage multiplication, and phage releases. The phage tail absorbed to the cell through a special receptor on the bacterial cell. The base plate and the fiber, if present, are involved in the attachment, and phage DNA is injected through the tail channel. Ca+2 ion are often involved in phage attachment to the host. The phage DNA then take over some cell function in order to synthesize new phage in an essentially parasitic relationship. Normal metabolism of the infected cell cases as the host first replicate its phage DNA and then phage protein. This process is called maturation, and with self-assembly of virion with the host cell initially had from around the viral DNA, followed by the attachment of tail. Finally, the lytic cycle is completely when a lytic enzyme, lysine, encoded on viral DNA is produced, resulting in cell lysis and release of infective phage particles into the surrounding environment. Lysine released from infected cell can lyse non-infected cell. The time from initial absorption to the release of phages is called the latent period. For lactococcal phage, this period ranges from 10 to 140 minutes. The number of virulent particles released per infected cell is called the burst size. This range from less than 10 to more than 300 for lactococcal phage. Lysogenic or temperate cycle. Lysogenic or temperate cycle. When a sensitive host is infected with temperate phage, two responses occur. The phage multiply like a virulent phage and causes cell lysis, or in a small percentage of the infected cell, the phage either integrated into the host chromosomes, prophage, or is maintained as plasmid in the cytoplasm. In both the cases, the phage replicates within the host cell and does not cause cell lysis. Lysogenic culture normally contain free phage without an effect on their growth or acid producing ability. Since prophage integration make the other cells immune to this and closely related phase, lysogenic immunity. Lysogeny is widespread in lactic acid bacteria. Pseudolysogeny and the carrier state. Pseudolysogeny and the carrier state. Pseudolysogeny and the carrier state. The simultaneous presence of both free phage and bacterial cell is called pseudolysogeny or the carrier state. When the phage is not integrated into the host cell chromosome and when only some bacterial cells are infected, but the culture apparently grow without any problem. Phage infection of host cell produce the following effect: retardation or slowness in acid production, slowness or retardation of flavor production, loss of production due to impaired starter culture metabolism. Source of bacteriophage. Source of bacteriophage. Phage strain against lactic streptococci may be found in the atmosphere of cheese factories. Finally divided particle of whey emitted from the whey separators appear to be the main vehicle for airborne phage contamination. Whey contaminated dust also play a part. Source of considerable phage contamination were the culture utensil and equipment and milk supply. The main source through which phage entered the plant is the raw milk truck and personnel entering the plant. The following factors enhance the incidence of phage attack in the starter: aeration of milk before inoculation, the use of large volume of culture, the use of stayed 4 to 5 days old sterilized milk. Phage detection. Phage detection. The fundamental methods are used in the detection of phage. The phage assay and inhibit of acid production. Control of bacteriophage. Control of bacteriophages. It is evident that existence of bacteriophage is a recognized problem in the dairy industry, and the following precautionary measures may be practiced. The propagation of the starter culture must be carried out employing aseptic technique. Heat treatment of bulk starter milk ensure the destruction of these viruses. It is vital that the starter tank must be filled to its maximum to avoid prolonged heating to eliminate the phage in the airspace. Daily rotational of phage unrelated starter strain and phage resistant strain must be used in the dairy. Effective filtration of air of starter room and the production area. The equipment must be properly sterilized. Location of starter room away from the production area and way handling department. Ensured that plant personnel, particularly those from the cheese room, are not allowed into the starter handling area. Propagates starter in a phage inhibitory medium. This is the medium deficient in calcium ion to prevent phage absorption. Do not use starter culture sensitive to those phage categorized by having short latent period and large burst size. Fogging the air in the starter preparation area and cheese production area with a solution of hypochlorite or alternative use of ultraviolet light can control phage. Development of phage resistant strain, use of mixed strain starter culture. Thank you.